So what led to the discovery of our modern cancer chemotherapeutics? The era of cancer chemotherapy began in the 1940s with the first use of mustard gas, a chemical warfare agent in which scientists at the United States Department of Defense, following autopsies of mustard gas victims, showed suppression of division of lymphoid cells, a non-cancerous cell type with properties similar to certain types of cancer cells. Mustard gas was too volatile and toxic to be used widely for the treatment of cancer, but subtle changes to the chemical structure of the molecule led to the generation of the so-called nitrogen mustards, a class of cancer chemotherapeutics which we now describe as DNA alkylating agents, because of their general mechanism of restricting growth, division and spread of cancer cells by physically bonding or alkylating with DNA. Although the nitrogen mustards were phased out many years ago, owing to excessive toxicity, this was the first step to the realization that cancer could be treated by pharmacological agents. Fortunately, cancer chemotherapy is now entering a new and exciting era. Advances in genetics and molecular biology has led to an increasing understanding of molecular processes behind specific cancers and the identification of a number of molecular processes and targets or proteins which are unique to a cancer cell. The design of agents which will act on these targets is resulting in the development of more selective anti-cancer agents with fewer side effects. An example of targeted therapy is the class of drugs we call kinase inhibitors. These drugs work on complex signaling pathways in cells by targeting and then blocking an essential protein involved in a specific function of a cancer. The first kinase inhibitor to reach the market in the early 2000s was Gleevec. This represented a milestone in anti-cancer therapy. It was the first drug to target a protein which is unique to a cancer cell. It acts as a selective inhibitor for a tyrosine kinase called BCR-ABL, which is active in certain tumor types. Since then, a number of kinase inhibitors have been developed for treatment of a number of different cancer types. Each inhibitor targeting a specific protein required for tumor growth, proteins involved in the process of cancer cell division, or proteins that are fundamental to the spread of cancer cells.